welcome back to our VLI Biology Online Labs. My name is Ms. Copay, and today we are going to do the DNA extraction, Lab 5. And so in your VLI Online Labs, this is Lab 5, and it refers to our lab manual, pages 79 through 81. So those are your references for the stepwise instructions that I'm going to introduce today. Also on Buzz, don't forget there is the lab report template that you're going to need to fill out about your purpose of this lab, your hypothesis, conclusion, and to also include pictures of yourself completing this lab. And then fill out the lab manual questions as well to submit into Buzz for grading. Okay, so DNA, our source of DNA for today is going to be our cheek cells, so we're going to be working with our own DNA. We could be doing this out of any source of living material. Common ones for extracting, and you could do the same thing with bananas. Bananas are a great source of DNA. They have like 8 copies of every chromosome compared to us. We only have 2 copies of every chromosome, so DNA and bananas is chock full of DNA in their cells. You could do strawberries. Other people do wheat germ. I've seen that done too. So we're going to be using our cheek cells today, which is pretty easy to do. What you're going to need in addition to let's start with the materials from the kit. You have meat tenderizer that you're going to be using. You're going to need your beaker here, and this is your test tube. So it may not quite look like a test tube, but it has a cap on it. And you're going to need that as well, and a dropper to transfer or pour your contents. Okay. Other materials that you're not going to get in your kit that you're going to need. This is isopropyl alcohol, also known as rubbing alcohol, 70% rubbing alcohol. Any dishwashing detergent that you have lying around the house. And then you're going to need a sports drink. So I'm using a clear one. It doesn't much matter, but I do think it's a little bit easier if you have a clearer one solution. So let's get started with our procedure. Again, the video that's on Buzz goes through this as well, so you may find that helpful too. Okay, so we're going to start with this, and I'm going to swirl this as a mouth rinse, and that's going to slough off any cheek cells that I have that are ready to come off. And remember that our bodies are constantly regenerating our cells. And so here we're going to be collecting those cells, because those are where our DNA is located, inside those cells in every nucleus of our cells. So I'm going to take a swig of this, and I'm going to swirl it around for a good 2 minutes, rinsing back and forth, you know, just to kind of vigorously. In fact, it's great to do this lab before you even brush your teeth in the morning, because all those dead cells will be there as well. And then I'm going to spit the contents into this container here, and this is where I collect my swirled spit, if you will, into this container. And then we're going to add to that, so let's do that first, and then we're going to add our soap to that and to break up those cells. So we'll talk about that as we go through here. So I'm just going to take a swig of it. Okay, that was lovely. So here contains my cells. It didn't taste bad either. Now what we're going to do is we're going to add a little drop of our soap into this cup here. And we're going to use our test tube here to transfer some of the contents. And I do this because then all of my reaction won't be in this test tube. We only have to fill it halfway with this. But it's nice because then if something goes wrong and I want to repeat it, I don't have to do the swirl again. And so I'm just going to transfer. You can pour it, or we can use our transfer pipette to transfer it. But I'm going to go ahead and pour the contents into here. Okay, and so I'm going to just save behind some so you can see I didn't pour all of it. I left some residual here. And now we're going to add just a couple drops of our Dawn dish soap. And so what's the purpose here? What are we going to use this Dawn dish soap for? The way that Dawn, our dish soap, cleans is by bursting cells open. And so this is a hydrophobic component in our soap. And when it comes into contact with our cell membrane, it breaks those cells open. And so it breaks the contents out. So it's going to open up our cells, and it's going to open up our nuclear membrane too, which encases our DNA. So by just adding a couple drops here, I'm going to break those cells open here. And now I'm going to swirl that around, give it a little bit of a swirl here. I don't want to make it really foamy. Okay, so I'm just adding that soap to break those cells open. So now this solution here contains your DNA, but it contains every other macromolecule, like our proteins and our lipids and our carbohydrates. All those organelles contents are in this solution here. we're going to use this meat tenderizer to break down the proteins. So remember I said that, you remember those 4 classes of macromolecules you learned about in Unit 1. Carbohydrates, proteins, lipids, nucleic acids. So our DNA is all we're interested in this tube, but all those classes of macromolecules are there. So our lipids were just broken down with our dish soap, and now we're going to add a pinch of meat tenderizer here. So this is a meat tenderizer, and what you think about that doing, its function is to break down proteins. So just like we talked about breaking down our lipids and our cell membranes, now we're going to break down our proteins. And I'm just going to add a little bit of the meat tenderizer and give it a swirl too. And now we break down those proteins. So now really all we're left with here is our carbohydrates. There's probably a little bit of sugar in there. But we're really left with our nucleic acids in this solution that are intact. So our lipids are broken apart and our proteins are broken apart, and hopefully our nucleic acids are here. So it tells you in your instructions to put your isopropyl alcohol in the freezer so that it's chilled. And this came out of the freezer and it is chilled. So it's going to help you to separate the nucleic acids from the rest of the cell components. And so when you think about our nucleic acids, these guys have a lot of charged molecules on them. And they like a water environment. They're nice and happy in solution, but we can't see them. What happens when I put this alcohol, which is a nonpolar molecule, in contact with this polar solution, this water-containing solution, is that our DNA, when they touch that alcohol, they coil up on themselves. They'll actually wrap themselves up and try and repel that alcohol around them, that nonpolar environment around them. And so we'll be able to actually see them. So it says to let this sit for about 10 minutes to break everything down. So I'm going to let it actually sit here for a minute. All right, so this has been sitting for several minutes now. And so what I'm going to do here is I'm going to create a layer on top. So this is our nonpolar alcohol layer coming in contact with our water, and we're actually going to be able to layer this right on top. And to do that, you want to drizzle on the side. And if you see, I'm tilting the tube here on an angle, so I'm increasing what we call the surface area that's actually going to come in contact with our alcohol. And so that's going anywhere that there's DNA in that solution, which is everywhere. It's going to actually touch that alcohol and then start to coil on itself or try to come out of solution on itself. And so if you look here, I don't know if you can see this very well, you'll see that a buffy zone is formed between the alcohol layer and the soap layer. And so that layer here, if we let it sit for a few minutes, I'm actually going to spin it in one direction, and that DNA will start to coil on itself. And you'll see a buffy layer here start to form. So right at that interface between the alcohol layer and the soapy layer on the bottom, you can start to see like strands of fingering their way out of the buffy, the soapy layer and into that alcohol layer. And so if I let them sit here, and if I twist it in one direction, you should actually start to see them come out of solution. If you can see those little strands of hairs they look like, that's your DNA. And so in every single cell in our body, we have over 3 feet of DNA coiled inside of the nucleus of every single cell in our body. So there's a tremendous amount of genetic material that you're going to be able to visualize. So I'm going to just sit him here, and I'm going to twist him around. And that DNA will twist on itself and create a nice ball of genetic material. So the longer you let it sit and kind of give it a little bit of circular momentum, it will start to wrap on itself. And so it says to let it sit there for 5 minutes, and it'll become even more apparent. So that's your lab for today, and hopefully you're able to see that nice DNA come out of your cells and what you're going to be collecting there. You can use your transfer pipette to actually suck up those contents and see them. You could put them on a slide and you look at them under the microscope even. Using a little bit of methylene dye, you can get great contrast and just see what DNA in your cells looks like. You won't see the A, T, C's, and G's. You're just seeing the overall chromatin inside your cell. Another thing you can challenge yourself with is use a Ziploc bag and put a single strawberry in it and add a little bit of soap and a little bit of water, a little bit of tenderizer, and mash it all together. And then cut a hole in the corner of the Ziploc bag, squeeze the contents out into a cup, and add your alcohol, and you should be able to see strawberry DNA too. So it's a very simple procedure that's used in labs all over the world to get DNA. So good luck with this, and if you have any questions, be sure and reach out to us. See you next time.